When it comes to talking about biomolecular structures, proteins tend to get most of the attention. But your nucleic acids, your DNA, your RNA, they have structures too. And now they're saying, I want to turn. So let's hear them out. This is going to be the first of two videos on nucleic acid structure. In this video, we'll go over the basics of nucleic acids, so DNA and RNA, what are they made up of? We'll talk about things like base pairing, as how the helixes are helixes and how their RNA is more flexible. But then next lesson, we'll get more in depth into the more complex structures, how they can wrap around proteins, how the bases can be modified, how mutations might affect or might not affect the protein that gets formed, these sorts of things. And so we'll talk about all of that in the next video. Today, let's focus on the fundamentals. Let's get some terminology down. Let's see what is inside of these nucleic acids. When you think about nucleic acid structure, you might think of that double helix. But that double helix is really just the beginning. And it's actually not the beginning. In fact, the beginning is going to be the actual building blocks of those nucleic acids that cause them to adopt structures like the double helix. And so let's look at the makeup of nucleic acids, so of our RNA and our DNA, and how that chemical makeup actually gives them the properties that gives them these layers of structure that we often don't really think about when we just see a string of letters like A, T, C, G. Those are referring to just part of the nucleic acid. They're referring to this part called the nitrogenous base or this nucleobase. And that's the part that allows for specific hydrogen bond formation or this base pairing that makes it so that you can use one strand of DNA or RNA to make a copy of another strand or make a copy of the same strand. We'll get into it. But bottom line, that's just part of this nucleic acid. And so let's talk about these parts. As we mentioned with proteins, a protein is a polymer made up of these individual monomers, these building blocks. And in the case of a protein, we had our building blocks were these amino acids. In the case of nucleic acids, here we also have a polymer, and our monomers here are going to be nucleotides. Similarly to how our proteins had a generic backbone and a unique side chain, our nucleic acids are going to have a sort of generic backbone, this sugar-phosphate backbone, as well as a unique, well, we don't really call it a side chain, but it's got this unique nitrogenous base or nucleobase. Looking a little more closely at these nucleotides, they're actually made up of smaller parts. We have a nucleobase, a pentose sugar, and a phosphate group or groups. There's a little bit of complex nomenclature when it comes to what we actually call these parts and what we call these partial parts when they combine together. Basically, DNA and RNA, they differ in a couple of ways, and the key way is that DNA has a deoxyribose sugar and RNA has a ribose sugar. You can think of this sugar as a sign of being the central coordinating part. I like to think of it kind of as a little person with two hands up and 2 feet. And the DNA, basically one of the right foot's missing. It's just the hydrogen. And in the RNA, it's got both the right and the left feet. It has OH groups on both of these. In terms of our naming positions, this right foot is going to be our 2' position. And so we can say that DNA is deoxy at this 2' position. The 1' position, this is basically where we're going to connect through what we call a glycosidic bond to our nitrogenous base. This 5' position, this is where we're going to connect to our phosphate groups. And this 3' position, this is what we're going to use to form these phosphodiester bonds to other nucleotides. In terms of that base, so we can call these nitrogenous bases, we can call these nucleobases. They're very weak bases, but they can pick up a proton, and so this can actually change their properties slightly if we have acidic conditions. There are 5 bases. There are 4 that we find in DNA, and 4 that we find in RNA, but the 4 that we find in DNA and the 4 that we find in RNA are almost the same, but one of them's different. They share adenine, guanine, and cytosine, but then DNA has thymine, whereas RNA has uracil. But as we'll see, thymine and uracil, they kind of act really similarly. They're both going to form base pairs with A. The difference is that thymine has this methyl group here, whereas uracil doesn't. And we'll talk later about why that is important. But in the case of these nucleobases, we can classify them into 2 different forms. We can talk about the purines and the pyrimidines. So the purines, these are the ones with the 2 rings, and the pyrimidines, these are these smaller ones with just a single ring. The way I remember it is pure as gold. So purines are adenine and guanine. And I remember that they have 2 rings because they're like pure, they've got it all. Whereas these pyrimidines, they're going to basically just have a single ring, they don't have it all. And it's not because they were like depurified from the purines or anything like that. This is just a little memory tip I use to remember that the purines have these 2 rings, whereas the pyrimidines have a single ring. We can see that these also, although these have 2 rings, they have a very different structure in terms of their opportunities for hydrogen bonding, and we'll look more at this in a minute. So as we saw before, the thymine and the uracil, they differ just by the presence of this methyl group in the thymine. And then the cytosine, it actually has an amine group up here instead of a carbonyl oxygen. If we think about our hydrogen bonding opportunities, well, here basically we have 2 acceptors and here we have 2 donors. So keep that in mind when we think about our base pairing. Sometimes when you see a sequence of nucleic acids, so one of those strings of A, C, T, G, you'll see that there will be parts that are either unknown or that they vary in different sequences. So people might use like a placeholder for the actual nucleotide that's present. An N is going to stand for like any nucleobase. So if you see an N in a sequence, that's basically can be anything. An R is going to stand for a purine. So one of our A's or G's. So the R, pure. And a Y is going to stand for a pyrimidine. So C, T, or U. So a pyrimidine. So we looked at our pentose part. So our pentose, we have 5 carbon sugar. And we looked at our nucleobase, our purines, and our pyrimidines. Now let's look at how we can connect them. As I mentioned before, that connection is happening at the 1' position on the sugar. So we talk about these prime positions, that's in reference to the sugar. And we're talking about here, we're saying, yeah, we're putting it on that right arm of the deoxyribose or of the ribose. And we're going to connect it to a nitrogen in our nucleobase through this dehydration or condensation reaction, which is forming this N-glycosidic bond. So N, because it's going to be through this nitrogen, we call this an N-glycosidic bond. When we do this, we get what's referred to as a nucleoside. So I like to think of this as a side of sugar. We have this S here, we have a nucleoside. If we add phosphate groups, that's when we get to a nucleotide. So I think phosphate, nucleotide. So the T is telling you there's going to be phosphate groups added. We can have 1 phosphate group, in which case we call this a monophosphate. We have 2 phosphate groups, we call this a diphosphate, or we have 3 phosphate groups, we call this a triphosphate. So for example, if we had ATP, this is telling us we have adenosine triphosphate. So where does adenosine come from? Well, remember how we said that the bases were adenine, guanine, cytosine, thymine, and uracil. Turns out that when we take them and we add them to the sugar to get a nucleoside, so our side of sugar, we actually change the name. The easiest ones to remember are with adenine and guanine. They go to sine, which makes sense because we've got sugar, we've got side, we've got sine. So adenine becomes adenosine, or in the case of DNA, deoxyadenosine, and guanine becomes guanosine, or in the case of DNA, deoxyguanosine. More complicated when it comes to our pyrimidines, so cytosine and Thymine are going to go to cytidine or deoxycytidine and deoxythymidine. So here we were already with an S, and now we're going to a D. And with the case of uracil, well, it's going to uridine. So these are changing to denes, whereas with the purines, they're changing to scenes. Talking about nucleotides, notice that we're using the nucleoside name because we talk about like a nucleoside triphosphate or a nucleoside diphosphate or a nucleoside monophosphate. Because if we said nucleotide triphosphate, well, that would be redundant and repetitive and redundant and repetitive. Basically, the tide, the T, is telling us it's got that phosphate in there already. And so if we talk about in terms of how many phosphate groups it has, we would need to say the nucleoside triphosphate. So we typically just say like AMP, ADP, or ATP. If we want to refer to any generic nucleotide, we can use the term NTP or DNTP. Going back to the idea that we can use this N as a sort of filler for saying any nucleobase. To review, we have this pentose sugar, this 5-carbon sugar. In the case of RNA, it's going to be ribose. It's going to have that 2' OH. It has that right foot. In the case of DNA, it will be deoxyribose. It won't have the OH group here. It'll just have a hydrogen at this 2' position. At the 1' position, our sugar is going to be attached to our nucleobase, our nitrogenous base, our purine or our pyrimidine. This is where we're going to basically get that A, T, G, C, or U. That's this part. And that's the part that's going to make specific base pairs. But that doesn't mean that the rest of it isn't important. And one of the other really important parts is going to be the phosphate group or groups. This is attached at the 5' OH position. And we can have variable numbers of phosphate groups attached. When these nucleotides join together, they do so by forming what we call phosphodiester bonds. So remember that an ester is going to be a carbonyl connected to an oxygen, connected to another carbon on either side. In the case of our phosphodiester bonds, basically phospho because it's got a phosphorus instead of a carbon, and diester because it's basically there's 2 of them. Remember that there's resonance in this phosphorus. So we could draw the double bond here, or we could draw the double bond here. So we could think of this as this diester bond, but it's a phosphorate version. So it's a phosphodiester bond. This phosphodiester bond is formed by basically when these nucleotides come together, they come together in their triphosphate forms. And then the energy from those phosphate bonds is used to join these together. And so this is how we can kind of pay the cost for reducing the entropy of bringing these nucleotides together. And the order in which we bring them together is going to be specified based on their sequence, because we're going to see that we have that base pair complementarity. You grow this chain in the 5' to 3' direction using the incoming nucleotides energy in order to pay the cost. As a result, we typically refer to DNA in the 5' to 3' direction. The 5', this is where we're going to have those free phosphate groups because that was the first one. It's not going to have anything to its left, so it's just going to have phosphate groups. At the other end, there we have the 3' end. And so here we're showing the DNA's 3' end because we can tell because it has this deoxyribose sugar here. But in RNA, we would just have an OH here, and we would have an OH here. So this would be our 3' end, and we can see that basically its 5' end is tied up. This phosphate group is attached to the one before it, but it has this free 3' position, which can now form a phosphodiester bond with an incoming nucleotide. If we look at the structure of nucleic acids, we can see that it kind of has these 2 sides. It has a side that has these phosphate groups sticking out. So we have this this alternating phosphate sugar backbone. These phosphate groups are going to be very hydrophilic. They're going to be charged. Water's going to want to hang out with them. What about these other side of the molecule, however, these nucleobases? If we look at them, they've got a couple of opportunities for some hydrogen bonding, but they're largely going to be nonpolar and hydrophobic. This should get your bells ringing. Hydrophobic effect. So water molecules, they want to hang out with other polar or charged things. They want to hang out with things that can offer favorable interactions, because water molecules have that high polarity where the oxygen is going to be partly negative and the hydrogen is partly positive. If we stick in something like a nucleobase that's hydrophobic, well, waters can't really form interactions with those very well. So it's going to want to cluster them together. If it had to individually hang out with each of the individual nucleobases, what this would mean was that the water molecules around each nucleobase would kind of have to get stuck in place. This is because they try to maximize their water-water interactions, and by doing so, they cinch around that hydrophobic thing, and this takes some water molecules out of commission into this sort of solvent shell. If, however, the water molecules kind of join together those hydrophobic things, so they push those nucleobases together, this is going then to free up water that would have been tied up, leading to an increase in entropy and energetic favorability. If we stick nucleic acids, therefore, into water, what's going to happen is that those hydrophobic parts, those bases, are going to be pushed into the center of the helix. Or wait, where did I get to? Helix? We'll talk about that in a second. But basically, those bases are going to be pushed together because of that hydrophobic effect. When these bases are pushed together, now they have the opportunity to form those nonpolar interactions, those things like our van der Waals and our dispersion forces, where we can have them temporarily sync up their electron clouds through chain reaction type events that are going to be individually weak but collectively strong, and these can sort of glue things together. However, we don't want to just glue things together any old way. That wouldn't make for very good transfer of genetic information, and it wouldn't allow us to form specific fancy structures. If we want specificity, now we've got to introduce some functional groups into the game. And in fact, this is what we do in the bases. So although the bases were mostly nonpolar, they have these opportunities for hydrogen bond formation. And importantly, each of these bases has different opportunities for hydrogen bond formation. So here I've shown the acceptors with this sort of pink, orangey, reddish arrow. So these would be our acceptors. So that would be like our oxygens or our nitrogens with a lone pair. Remember that a hydrogen bond acceptor has to have a lone pair. And then we have the hydrogen bond donors, which are going to have hydrogens attached to something electronegative. And so in this case, we are going to have our donors be in these amine groups. So you can see that guanine here in this, if we look at this face, we'll talk in a second about how this is called the Watson-Crick or the WC face. And here you would have an acceptor, a donor, and a donor. And it corresponds nicely to cytosine's WC face, where it's got a donor, an acceptor, and an acceptor. Similarly, adenine, well, here we have a donor and an acceptor. And thymine, we've got an acceptor and a donor. And uracil, well, here it also has an acceptor and a donor, because that methyl group on thymine is on the other side. So these are going to pair up nicely. You'll see in each of these cases as well, we have that one of the big ones, the purine, is pairing with one of the small ones, the pyrimidine. This is going to make it so that you have a constant width if you have these bases pair with one another. What you might also notice is that things on one of these sides are kind of flipped. In order to form these complementary pairs, one of the strands needs to be running in the opposite direction of the other. So that is, the strands need to be anti-parallel. In an anti-parallel strand, we have one strand running from 5' to 3', where remember the 5' refers to having a free 5' phosphate group. And then the other is 3' to 5', where that 3' has the free OH group. And so here is just showing you a part of the sequence, but at the end of this, you would have that phosphate group, and at the end of this, you would have that OH group, and at the end down here, you would have the phosphate group, and at the end down here, you would have the OH group. In these cases, what we're seeing is that we're seeing the base pairing between these anti-parallel strands. So we have our A to T, or in the case of RNA, we have A to U, and then we have our G to C. There are a couple of things that you might notice here, and one is that we have these 3 hydrogen bonds in the case of our GC, and the 2 hydrogen bonds in the case of our AT. However, that is actually not the reason why we say that GC regions are stronger or that they have a higher melting temperature. Instead, the additional strength of those GC regions, the fact that it takes more heat to pull them apart to denature strands, comes from base stacking. So we're looking here at base pairing where we basically have specific hydrogen bonding between the bases, between different strands. Well, we also have this thing called base stacking. Base stacking is where we have the bases in the center up here that were pushed together, remember? Well, now they're going to be syncing up their electron clouds and kind of forming these interactions with one another. So when we talked about all those induced interactions, all that van der Waals, all that good stuff, that's happening between the bases in this up and down orientation. If we go back and we look at the structure of these bases, what we'll see is they're actually aromatic. They have their electrons delocalized above and below the ring in a pi bonding system. When you have these delocalized electrons, this whole kind of big glob of electrons above and below the plane, well, now you have this large region that's perfect for forming these sorts of nonpolar interactions, these van der Waals, these dispersion forces. In order for them to optimally kind of sync up their clouds, however, they have to be in the right orientation. It turns out that GC-rich regions have a more preferred orientation. They can form better base stacking interactions. So if we look here at the delta G of stacking, so basically if we put a GC on top of a GC, or if we put an AT on top of an AT, we can see that if we were to put a GC on top of a GC, we would get a negative 2.17 kilocalories per mole, negative telling us this is favorable, so this would be highly favorable. With an AT-AT, here we're still getting favorable interactions, but it's not nearly as much. Here we're just getting about negative 1.34. Therefore, if we wanted to pull apart these strands, we would have to put in, well, the opposite amount of energy. We'd have to put in more energy in order to pull apart the GC-GC than the AT-AT. And in terms of energy, we often think about heat. So we can talk about the melting temperature as the temperature at which DNA strands are going to pull apart from one another, or RNA strands are going to unfold. Basically, when we have the structure denature or like kind of melt away, that would be our melting temperature. And if you have GC-rich regions, this is going to be higher. This comes into play in the lab when we do things like PCR, where we're trying to replicate DNA strands. We have to melt, we have to raise the temperature to separate the strands, and then anneal. So basically bind little pieces of DNA called primers onto those sequences. And the temperatures at which we do those various things is going to depend on the GC content of our nucleic acids, with GC-rich regions leading to a higher melting temperature. And remember that that's because of the base stacking interactions and not just because of that hydrogen bonding. It's easier to think, yeah, GC, you've got 3 hydrogen bonds instead of 2. But remember that the hydrogen bonding is more about the specificity than the driving force of the binding, which is going to be our hydrophobic effect. It's going to be our base stacking, keeping things stabilized. Whereas the hydrogen bonding is kind of determining where these interactions are going to be made, giving us that specificity. Also, although these hydrogen bonds aren't the key force of the binding, they actually do offer more stability than we would see with other types of hydrogen bonds. This is because they're optimally arranged. They're oriented in ideal positions. So remember, we wanted to have that linear arrangement of our donor and our acceptor and that short distance. That's going to make them strong, as well as the fact that they're in this hydrophobic environment. Thinking back to when we talked about Coulomb's law and how a salt bridge in a lipid environment would be stronger because there's less polarity kind of interfering with that. Well, here we have these hydrogen bonds in this nonpolar environment, this hydrophobic environment. This is going to strengthen them as well. So we have this base pair specificity, where we're going to have our adenine to our thymine, or our uracil, and our cytosine to our guanine. And so this is our AT and our CG. And this makes it so that we can use one strand of DNA to copy another strand of DNA, or use a strand of DNA to copy a strand of RNA. Because we can have this base pairing relationship, this is going to make it great for passing on genetic information. The aromaticity also had another effect in that it makes it so that DNA is going to absorb UV light of around 260 nanometers. So when we talked about protein, we said it absorbed most strongly at 280 and 230. With DNA, we're going to see absorption at 260, as well as with our RNA. And there are actually different ratios you can use to kind of figure out how pure your DNA or RNA is. But let's go back and let's think about the structure of our double helix. So we have those anti-parallel strands of DNA, where the kind of order in which they glob together is going to be determined by having a complementary sequence. And then the globbing together is going to be driven by the hydrophobic effect and held together by that base stacking. How do we know about the structure of the double helix? Well, for that, we have to thank Rosalind Franklin and her grad student Raymond Gosling, who took the diffraction image showing the double helix structure of DNA. So this is similar to X-ray crystallography, except that it's actually fiber diffraction. So these aren't actually DNA crystals, but fibers. And when they did this X-ray diffraction, they were able to get this characteristic fuzzy X that gave them information about the spacing and the pitch of the helix, as well as the fact that it's double-stranded. The reason why I was kind of particular about, this is a fiber and not a crystal, is because DNA tends to crystallize in this artificial form, as far as we think, this A form. It's more kind of compact form. Whereas instead, in our bodies, DNA typically exists in this B form, this kind of more expanded form. Both the A form and the B form are right-handed helices. So if you put your right thumb up and trace the movement of your thumb, trace the flow of your fingers, that's going to follow the chain of this helix. In the case of A and B, they're both right-handed. There's also this thing called Z-form DNA, which is left-handed, but it's thought to be pretty rare and only forms in like certain sequences and things. We're going to focus mainly on this B form, as well as with RNA, we'll see if we had double-stranded RNA, that typically adopts the A form. With these DNA helices, you can see that we kind of have this major groove, this bigger part, and this minor groove, the smaller part. What we were looking at here was kind of what's available to bind. So not all of the acceptors and not all of the donors are going to be tied up in hydrogen bonding with the other base in its pair. Instead, we're going to have options still open. And these open options are then going to be able to interact either with other nucleic acids, with other proteins, with all sorts of different things. And so there are opportunities for interactions. Let's talk about some of the interactions, not only with proteins that we could form, but with bases as well. This especially comes into play in the case of our RNA, which is more flexible and tends to be single-stranded, but fold up onto itself. If we think about these bases, they can actually rotate. They're not held in place as tightly as we had with like our peptide bonds where things couldn't rotate. Instead, we can have rotation. In the case of our purines, here we can see what's called the syn form and this anti-form. You can think of it kind of like twisting a flag. We can get this syn and the anti-form in our purines, but we typically see the anti-form, and we typically only see the anti-form of our pyrimidines, because here, if we were to rotate things, this carbonyl oxygen would clash with the rest of the sugar. But we can get these flipped forms of our purines. So remember, pure as gold, adenine, and guanine. When we have these flipped conformations, well, now we can actually get different base pairing interactions. So our canonical interactions were going to be our Watson-Crick or our WC interactions, and they were happening on this WC phase. But we also have this additional phase, these Hoogsteen positions. The Hoogsteen positions, these are going to involve the nitrogen in the smaller ring of your purine. So they're going to involve the nitrogen in this 5-membered ring. I like to think of it as kind of like the smaller or less important base pairs, sorry, Hoogsteen pairs, but they're more of a minor contribution to nucleic acid structure. These are going to involve this smaller ring of the purine. So this nitrogen on this ring, when you're trying to remember which side it is. So we have this nitrogen in this ring, and then also still that same nitrogen that was contributing to as a donor before is still contributing, but now instead of looking at this face, we're looking at this face because we flipped this from an anti-form to a syn form. And now we're able to form kind of alternative interactions. In this case, we're still doing an AT, but with some cases, especially if we have protonation or deprotonation, now we can actually form different interactions. So we can get these non-canonical interactions at this Hoogsteen phase. We talk about this mostly in the case of our RNA, where we can form all sorts of interactions with the RNA, but we can also form these in DNA. And this leads to things like triplex and quadruplex formation. In the case of RNA, it can form those Hoogsteen interactions, and it can also form other sorts of kind of non-canonical interactions. One of the most common is a GU. We call this a wobble base pair, where we have this GU pairing, and it comes into play when we talk about mRNA translation, where you have a codon and anticodon pair. They're often going to have a GU. These aren't going to be as strong as the GC interactions or even the AT interactions, but in the context of a stretch of RNA where there's stronger bonds on either side, then these wobble pairs can exist. Additionally, RNA can form interactions through its 2' OH position. Remember, DNA only had a hydrogen here, but in the case of RNA, well, now we have an acceptor and a donor. We can form additional interactions here. And this can lead to complex folded up structures where we can not only have all these different positions on the base interacting with things, but we could also have these positions in the ribose sugar itself. And this is going to lead to all sorts of different structural possibilities. And when we talked about protein structure, we talked about layers of structure, how we had that primary structure was just a sequence of amino acids that folded up to form these backbone interactions that gave us our secondary structure. We had things like alpha helices and beta sheets and strands. Then though we had our side chains get involved, we got this tertiary structure. And then we kind of had protein chains interacting with one another to give us quaternary structure in some cases. In the case of nucleic acids, our primary structure is still just the sequence. Now, our secondary structure is going to be base pairs formed within a single strand. So this can be from things like hairpins. So we have these kind of local twist turns in. We can talk about kind of having this end part and then this stem. So when we have a stem, it's kind of this more linear part, and then we have the turn at the end of our hairpin. This is an example of a secondary structure. We can also have tertiary structure. This is going to involve longer range interactions, typically facilitated by loops and bulges. So we can get internal loops and bulges. A bulge is kind of like a little part that sticks out that's not paired, and then a loop would be a sort of bigger region. You can imagine you can have loops and that are basically bringing parts of RNA from one part of the RNA to another part of the RNA, and you can get interactions between these. And because you have all those different donor and acceptor sites, you can get these complex interactions. These would be examples of our tertiary interactions. We're within this single strand, but we're within kind of parts that are coming together from multiple regions of the RNA sequence. If we have that RNA then interact with another RNA or interact with a protein, we can call that quaternary structure. Bases can also be modified. We'll talk more about some sorts of like unintentional modifications, but there are also intentional modifications, and you see a lot of modified nucleotides in the case of RNA, especially in things like transfer RNAs and ribosomal RNAs. There's a lot of modifications. We can see things like methylation, as well as sorts of alternative bases. We talked about pseudouridine, which kind of had uridine, but it was attached at a different position. So there are lots of different options that are then going to lead to even more RNA binding opportunities. So you can see in this structure, for example, here we have a 7-methylguanine. So we have an additional group. We have all sorts of structural opportunities in the case of RNA. But with DNA, typically you just have that double helix. But that's not where DNA structure stops either. DNA doesn't want RNA to have all the fun. Instead, it says, hey, RNA, I see your quaternary structure, and I raise you my quaternary structure. I wrap around histone proteins. To review, DNA and RNA, they have structure. It's not just the primary structure, not just the sequence. When we think about DNA and RNA, it's easy to think about just a string of letters, but those letters are actually these building blocks, these nucleotides, which consist of that sugar, that pentose sugar, ribose in RNA, and deoxyribose in DNA, attached to a nucleobase, so one of our purines or our pyrimidines, and attached to phosphate. And these are linked together through these phosphodiester bonds. When they do so, you get these strands that have a hydrophobic part, the part with the bases, and the hydrophilic part, that sugar-phosphate backbone. In order to satisfy water, those bases get pushed together. The hydrophobic effect drives them together, and then once they're driven together, you can get those base stacking interactions where you have these transient charges, these van der Waals dispersion forces that are going to hold together everything in the center. But actually, how do we get the specificity? This is coming from the specific base pairs that form between the different bases. So we have G to C and A to T or A to U. These are happening between anti-parallel strands or anti-parallel regions of a folded up strand. This makes DNA really great for storing genetic information and passing it on because we can use one strand to make a copy of the other strands. DNA tends to form this sort of B-form double helix, whereas RNA tends to be single-stranded, can also fold up into itself with complex structures. So we have this kind of like stem loop structures. We've got these internal loops and bulges, as well as parts that are going to be single-stranded, but then those are typically going to be interacting with things because, well, everything likes to interact, right?